I have the honor to welcome you to this, the honor and pleasure to welcome you to this, I have to say. This is a workshop which we are now co-hosting between 3 Horizon Europe projects, that is iCare, Nanopass and MACRAMÉ, and we are obviously throughout what we are doing in this workshop supported and working with the Malta initiative. That is why you get this a very busy first slide, but fear not, I will tell you a little bit more about the projects in a minute. This is 3 projects of the 5 projects that are working together. We are a total of 5 projects that were starting pretty much around about last year this time. Some of us started on the 1st of December last year, and a few more then at the beginning of the following year. We have AcumenIST as one of the projects that is following this in alphabetic order that responded to this call and was awarded. iCare is one of our co-hosting projects today. MACRAMÉ is the one that is giving you the pleasure of this fabulously colored background, including the pink logo, which will stick with you and you won't forget after you stared at this for 2 days of workshop, I can guarantee you. Nanopass is another one of our co-hosting projects today, and Potential is the 5th and last but definitely not least in this club of 5 sister projects. I give you a quick overview as to what we were actually asked to do by the call text, but then obviously every one of these projects has a slightly different take on, oh gosh, going backwards instead of forwards. That's a good start for the workshop. Hang on, I'm trying to fix that. There you go. So every one of us responded to these expected outcomes, and we all obviously, like it is with these projects and sister projects, we have a slightly different take on what was asked from us, but in one way or another, we are addressing these 6 expected outcomes or impacts across the 5 projects. A lot of effort is done to develop high-resolution imaging methods, specifically quantifiable ones for use of the characterization of nanomaterials. And some of us have actually, including the project you are seeing right now, the MACRAMÉ project, we have advanced into the area of advanced materials. So we are building on our nanomaterial expertise that we have grown over the last few years, where we are now looking at a much broader range of materials. Many of our projects that you can see here, the 5 that are working together now, are actually addressing graphene as one of the 2D materials we are now moving into under the advanced materials label. A lot of that has to do with developing validated protocols. A big part of what the next 2 days of workshop will have to be about. We are also developing control experiments to relay in vitro models to address current gaps in toxicology on the materials we're looking at. We are delivering reliable data and improved data grouping guidelines, computational modeling, doing a lot in data storage and much better data handling to be shared across a project internally, and now also working in hopefully sharing data soon beyond every one of the projects and between the projects. A lot of what the discussion over the next 2 days will be about is on the bullet point number 5 here, and that is the harmonized standardized test methods. And obviously, all of this is looking at working on real world materials. So we are looking at increasing efficiency and effectiveness of what we are doing with regard to materials and product development. All of that you can find on our separate websites. You had the link of those on the previous slide. The slides will be made available to you via the website that you're already familiar with. And if you're not familiar with that, I encourage you to go there, because that is also where you find the latest version of the agenda. My task here today is to welcome you and to introduce the agenda to you. And the latest version of that you find always on the website. And we are demonstrating that by having actually changed the agenda right this morning, as we heard that one of our speakers unfortunately is ill. So go to the meeting webpage in order to download the latest agenda. We have unfortunately lost Andrea Haser of our esteemed speaker list this morning. She is ill. We wish her getting well very soon. But what we have done is we have stretched out the talks a little bit, meaning we are not in very much of a hurry this morning, but that is not an encouragement to any one of the listed speakers to speak an additional 20 minutes to the scheduled ones. But what we would like to do is we would like to go into the lunch break a little bit easy, having a little bit of time, but then start sharp again at lunch at 12:40. What you will get to see throughout the entire workshop is we are rotating this chairmanship and this welcome and the guidance of all of this. So today you're going to have me in the background and me welcoming you right now. And tomorrow you will have our sister projects from iCare here speaking, and Esther from INL. Sean is representing our sister project at iCare and NanoPass as well. So we are essentially rotating the very warm welcome that we are extending to you to be part of our joint workshop today. And with that in mind, what I will leave you for a moment just now is something that gives you a little bit more of an overview of what else these projects are doing. If you like, you can take a picture of that, but you can at any one time come back to the website and find more information about these two meetings. One of them is an in-person meeting we are running next week in Berlin, kindly hosted by Bauer. And the other one is a longer shot of actually working together on a joint workshop during what we are currently calling Materials Week 2024. And we are already reserving some dates for that in Limassol in Cyprus. But now is about your harmonization and standardization workshop, and that leaves me to say thank you and to hand back over to the stop sharing, to the running agenda for today. Thank you.
Thank you ever so much, Steffi, for that introduction. And we started on time, and we've actually come up slightly early, so we've even gained some more time. But it gives me great pleasure at this point to introduce the next speaker. Unfortunately, she's been having some technical difficulties this morning, so I have her slides to share. So I'll just have to quickly load those up whilst I make the introduction. So it gives me a great pleasure to introduce Mar Gonzalez from the OECD. Many of you, I'm sure, will know Mar, but she's very welcome at this workshop and has an important role to play in the whole test guideline process. And I'll hand over now to Mar, who I believe is joining us via her phone. So I'm not sure if she'll be able to actually share her video.
I'm not able, and can you hear me?
Can indeed, Mar.
Oh, at least that's working. Thank you. And I apologize for this. It has been like an IT challenge day for me today. But thank you so much for the invitation. I mean, already I apologize because I think I mentioned MACRAMÉ somewhere, but even in the title of the slides. But I want to thank you, all the projects, for giving some space for the OECD to present what we're trying to do at our end. And hopefully this you will find it interesting, and there are some opportunities as the projects move forward in collaborating a little bit more. So I also apologize for those that already know me, because there are some slides that you are familiar with, but just in case there are new people here, I just want to go back to the basis of where I'm coming from and what is the work that we're trying to achieve at the OECD. So if you can go to the next slide, Sean. Thank you. So one of the main purpose of the work that we're trying to do at OECD is to develop some standards that respond to some regulatory needs. And we have in place an international agreement, which is called the Mutual Acceptance of Data. That it mainly means that data generating using the OECD test guidelines under good laboratory practices are accepted across member countries and MAD-adhering countries having the same data requirements. So I think I lost, yeah. So basically what we're trying to do, the OECD test guidelines are these single quality standards that should be applied for testing all chemical substances. And this is where we have tried to incorporate as much as possible nanomaterials, being them chemicals. The rationale behind is at the end that you can test only once and that the results from that test are accepted everywhere. And the success of the system really relies on regulatory and scientific acceptance. And from a regulatory point of view, what we will understand by regulatory acceptance is that we have a clear overview of the regulatory requirements in countries for biochemical legislation, that there's a clear regulatory need for an alternative assay. We have to specify, for example, whether what is the intended use of the assay or battery of assay. It is a replacement, it is an alternative, is it for hazard identification? And another thing that needs to be considered is that the accessibility of the assay. So it should be possible for several labs to perform this. So we have to have an early discussion between methods developers and regulators to try to have like a solving attitude, if you want, as we move forward. Then the other element that I mentioned is the scientific acceptance. So it's really the clarity of the experimental data, so the positive, negative, challenging chemicals, clarity of the applicability domain covered, or their limitation, for example, or is there sufficient data to justify the intended applicability domain? And here is where, for example, some of the projects that have been mentioned before is crucial, in that if we don't have mature research data, then we cannot start at the OECD level. So we have to have predictive model, and at the end that it has to be reliable and that it has to be repeatable in several countries. So that's basically like the background to the work that we're trying to do with the MATS system. So if you go to the next slide. So at the OECD, we have two main work bodies, if you want, official bodies that work on the safety and standardized methods for nanomaterials and now advanced materials. So it's the Working Party on Manufactured Nanomaterials that I'm leading. And the idea here is really to have all this policy discussion on innovative materials. Okay, I think, yeah. Kirsten, can you see the slides? I can see the slides, so I don't know if it's a problem for everyone.
I can see the slides, Mar, no worries.
Okay, excellent. And what we really try to do at the WPMN is to have all this discussion about what could be the needs and try to anticipate what are the areas that will need to be covered. And when we have some more clarity on this, then if there is a test guidelines or a guidance document that needs to be developed, then this is submitted to the Working Party on the national coordinators of the test guidelines program. And here we have like joint expert groups that develop the test guidelines or the guidance documents as needed. At the end, what we really want to achieve is regulatory acceptance of the methods. Next slide. So thanks to some of the project as Nano Harmony, for example, or Go for Nano, we have been able to move forward in developing a number of documents that have already been published on nanomaterials. So you have them on the screen. I will not repeat them all. But I think something to note is that really like the Malta initiative and the fact that there were some specific funding for developing of these test materials have really made the before and after in standards for nanomaterials. As I said, we rely very much on data. We rely very much on the research and the maturity of the research. Otherwise, we cannot start a project within the OECD, or we stagnate, actually, which has happened in the past. Next slide. There are a number of ongoing projects that we're still trying to implement. So there are several on FISCEM properties, as you can see on the slide. The most advanced one is the one on surface areas. on determination of solubility and dissolution rate. So we have already started the first round of comments. There are still some sections in the guidance document that needs to be further developed, but we are hoping that we can submit that for approval in April and then to have it declassified by June. And then some of the other projects, I think there will be more, it's more likely that they will be finalized in 2025 or 2026. So all these projects that you see on physics properties, biotic systems, human health, and fate and behavior, those are done by the test guidelines program. So there are the 4 sections that are addressed on the testing methods. But all this work is supported by other guidances done within the Working Party on Manufactured Nanomaterials. Some of the guidance that we are working on right now, for example, is the section 6.9 on grouping, which is part of a larger document for chemicals, the guidance on grouping GD 194. And we have practically finalized the section on nanomaterials, and we hope that the whole document can be published in 2024. We had another document that is currently being reviewed, has been finalized and is in the process for declassification, which is a scoping review to consider a tier approach to accurately determine the bioaccumulation potential of manufactured nanomaterials. And this is one of the examples where you can see that where we're looking really at the state of the art on the information that we could use for a later proposal for a test guidelines or a guidance document to support testing assessment of nanomaterials. Germany, for example, is developing a guidance on release test for manufactured nanomaterials. And another one that is that we finally managed to push forward, and we hope to have something more concrete in 2024, is the guidance on sample preparation and dosimetry. And this is one of the most important documents, perhaps, that we have drafted, because it's like the document that you need to follow before you even start to use the test guidelines and the guidance document that have been prepared. So this is something that is coming up, and I hope that we can have for you in 2024. Next slide. So I do want to mention that, I mean, we have a very specific, very timed process for the development of test guidelines and the submission of new projects. And we just completed and received all the submissions, and we received one on nanomaterials for the section on human health. And this is to develop validating the in vitro micronucleus assay for engineered nanomaterials. The leads of these projects is the UK, Germany, France, and Norway, but we also have some partners from the US and Luxembourg. And I think if I'm not mistaken, Tommaso is around and is one of the partners of this project. And this project is a follow-up of the study report on preliminary guidance on the adaptation of the in vitro macronucleus assay, which include an SOP. And the idea is to use this SOP and to try to do some interlaboratory trials and see how much needs to be revised on the guidance document 349 and the test guidelines itself. So this is something that is going to be discussed in April, and hopefully will be approved and included in the program of work of the test guidelines program. Next slide. So Steffi mentioned already that based on the expertise of nanomaterials, we are all enlarging the scope or the materials that we are covering. I think for the nanomaterials, we know that we have achieved like the safety of nanomaterials is now better understood. We also acknowledge that there are still several areas for which new tools need to be developed. Some of the areas, for example, that have been mentioned is the dispersion potential of nanomaterials for human health testing. And most of the documents that we have developed to date are really on first generation nanomaterials, so silicon dioxide, titanium dioxide, etc. So we we acknowledge in the WPMN that more efforts will still need to be done to develop the tools to assess those nanomaterials, but that we also have to start focusing on tools for multi-component nanomaterials, advanced materials. And with this in mind, we started some work on and to start to think how to address on what could be the best strategy for advanced materials. And I think one, if you want, we can characterize it that is like a two-way direction. So first, it was to develop a strategic approach. So it was really to develop a tool that can help us to screen new materials and to identify specific actions needed. For example, whether we need a test guidelines, a guidance document, any standardized methods that are missing for specific advanced materials. So we can incorporate them in the regular work that we have at OECD. But at the same time, there is also acknowledged that all these test guidelines and the standards need quite some time. The validation process is quite lengthy and needed and crucial for the mutual acceptance of data. So we are trying to develop also some ways to start thinking about the safety and sustainability at the early stage of innovation. And for that, we have established a work area on the safer and sustainable innovation approach, and we have had already some publications and webinars that you can assess if you want to look at. Next slide. So for the strategic and the tool on the early awareness and action system for advanced materials, which is the early for ADMA. I don't know if you can go to the next slide, Sean. So we started with a couple of case studies that were mainly focused to review the applicability of the strategic approach and to make sure that this early for asthma system could be fine-tuned with real case studies. Next slide. I think I missed one slide. No, that's fine. Then we had, we fine-tuned the early awareness and action system, this ADMA system. So we published this in September. And it's really a tool in anticipatory risk government approach for timely decision making. It's mainly targeting audiences, risk assessor and regulators. So you already need to have a certain amount of data to use this tool. And you really rely on existing data and expertise assessment. The report has been published, and it also includes an Excel tool where you can try to use your own case study, your own material, and try to answer the question to give you a quick overview about the areas where there is some data gap. We invite you, obviously, with the materials that you're working on to use this tool and to get back to us if there's any suggestions to make it a little bit more user-friendly or if you have any difficulties. I mean, the idea is that this tool can be further refined if we see that it's needed. The idea is to make the life easier of the user and not to make it more complicated. So it's really should be easy to use and quick to use. Next slide. So now that we have this early for ADMA tool, what we're trying to do is to develop some further case studies. We already have one on nanocarriers and silver cellulose articles with Asina. We have the report under review, and hopefully we can make it available to you early January. We have also partnered with Sunshine to do another case study on graphene oxide. And now we have a series of case studies that have been organized, one on 3D printing in June 2024, and one that might be of your interest, which is on the graphene-based material. So the idea is really to have some case studies to use the early for ADMA and to have some specific recommendations on what could be whether there is a need for specific test guidelines, for specific guidance, or any other tool in there. So if you go to the next slide. So this is just to provide you with an example on the learnings from the nanocarrier case study. One of the challenges that we have with nanocarriers is the plethora and the variety of nanocarriers. So we are already thinking about having a follow-up workshop that we can go into more in-depth on some of these specific applications. But the idea from this first nanocarrier workshop was really to test whether if the application is different, how much difference will be also using the early for ADMA. But you can see already that there is the need to establish harmonized test methods and analytical method. And this is one of the areas where we're going to be focusing on a follow-up workshop. Next slide. So just to conclude, I mentioned the graphene-based material, the case study. So we are having a first call in December, so we are still doing a doodle, but the idea will be the first call is really to review the inputs that can be provided by contributors. So we have several countries that already raised their interest, like Spain, Germany, Netherlands, South Africa, and we have also much contact with the experts involved in the graphene flagship. And here I'm missing Sweden, sorry. We will have like a first online meeting to advance the materials that will be circulated in advance to the workshop, and that is expected to happen in February 2024. And with the view to have like a larger workshop in September, October. So still, as you can see, I mean, we're still discussing the deadlines and the timing for this event. But perhaps this is an area where the input from these projects could be incorporated, and where obviously you have an extended invitation to participate on this. And with that, I think I just, if you can go to the next slides, it's just, the slides will be, I guess, provided. So it's just to give you the links to the task guidelines and guidance documents that have been published so far, and also access to the webinars that have been done to present the scope and limitations of each of these documents. And then the next slide is also for the safer and sustainable innovation approach, if you are interested. And I think that's almost the last slide, which is a thank you, I think, at the end.
Thank you ever so much for that presentation, Mar, as ever, and for continuing to support us in all the work that we do. We have plenty of time for questions. If anybody has any direct questions they'd like to ask Mar, if you just raise your hand, and then we can direct the questions to Mar. Maybe then, Mar, it probably then falls to me as chair to probably ask you a question, and it's just really kind of around how useful have all of the EU projects been in terms of developing new methods and directing them towards the OECD? Are you seeing sort of more new methods coming through to test guidelines because of these?
Honestly, I think has been instrumental to have the EU projects developing all this data. I mean, we have seen like, for me, there is a before and after the Malta initiative and all these projects coming into place, because there is, it has speed up, if you want, the process. And one of the reasons it has speed up is because there is more clarity on the proposal made. So there is more clarity about the modification. There is more clarity about what is test methods that are needed. There is much more experience about what is the type of answer from a regulatory point of view that you want to answer. So for us, this from the OECD point of view, I think this has really made a big difference.
Thank you. So we have a question from Irene. If you can just unmute yourself and ask. Thank you.
Good morning. Thank you. Thanks for the very interesting presentation. And I'm very interested on the mutual acceptance of the data. And I was wondering if if we have already something like a sample from this initiative, or when shall we expect the first fruits, the first data set, let's say, because you did mention that it's most important for computational approaches. This is the first question. And the second one is, if OECD holds data sets from European projects that have been done, and if so, who is the contact person, the contact point to get the hand on those data?
Okay, so there, I think the first question was more about, you were asking about the mutual acceptance of data. And I'm not clear about your question. The question is, once we receive the data, how much time it takes?
The first question was if this started happening right now,
Right.
or we already have the first fruits of this mutual acceptance of data. If we already have, for example, data sets that we can see the tests that have been done, so we don't have to measure them again.
Yeah. So the thing is, like, I mean, you have data that has been done at the research level. Okay. So the process will be that you need to make a proposal to the OECD. You have, in this proposal, you have to explain the data that you will be using, the regulatory question that you will be answering, or whether this is to decrease the number of animal use, etc. So you will be submitting this project proposal to the OECD, okay? The more data you have, the more clarity and the more the more you describe the type of modification or the type of guidance that you want to develop, the easier it will go. Okay, and I think for that the best approach. I mean, we can have a discussion, bilateral discussion to explain you that. But there is also NanoHarmony that has been recently published their mentoring tool, which has been that is extremely useful because it gives you a very clear explanation of the different steps that you will need to follow. And it's only once that you have finalized a project within the OECD as a test guideline that this test method is part of the mutual acceptance of data. Okay, so we have two types of products at the OECD, if you want, on testing, which is like the test guidelines, which are really like the recipe that are part of the mutual acceptance of data, that are part of regulation. And then we have guidance documents that are more simple document, if you want, because you don't, it's based on the available information, but it doesn't need all this validation exercise that will be required for a test guideline. And those are supporting documents, and they are not part of the mutual acceptance of data. However, a guidance document can tell you how to use an existing test guideline and how to make the necessary adaptations for nanomaterials. So that's how it works. So really, for the mutual acceptance of data, you really need to have the test guideline completed. And then it is for the different countries to decide in their regulation which test they're going to ask a company to submit. But what they cannot do is to ask this company, if they follow the test guidelines and they are done under good laboratory practice, what they cannot do, the country cannot do, is to tell them to repeat the test. But that doesn't mean that the country can say, I mean, this product is not approved. So it's really about the testing solely. The mutual acceptance of data is only about avoiding repeating tests. It's about trust in the test, yeah.
I understand about avoiding doing the test, but will the user, for example, get back the measurements already done
I think, Irene, I think there's maybe a little misunderstanding here. The OECD, when it comes to test guidelines, the test guidelines are developed using the OECD process and accepted by OECD countries. But the data that a company would produce is then sent to the appropriate regulatory body of the country in which they are looking to go through that regulation for. So, for example, the OECD doesn't receive the data from all the companies. The companies will submit their dossiers to, for example, in Europe, they'll be submitted to ECHA. So the OECD is just merely setting the sort of parameters of the test guideline rather than necessarily administrating them, so to speak.
The mutual acceptance of testing, let's say.
Yeah, so the mutual acceptance of data is that if you produce, if you produce data using a test guideline, you can reuse that data in another country to also comply with their regulation. So if you're submitting a product in the US and the EU, you can use the same data without having to repeat it. Just for yourself and everybody else, in the chat, there's a link to the NanoHarmony Process Mentor, and that explains a lot of the whole process, helps guide or people developing new tests through the process. But there's also the link in there to some training materials, which gives a lot more sort of background on OECD test guidelines. But if it's okay, I'll switch over to the next question because we now have a few questions coming in. So, Jacek, I believe you're next.
Thanks. Thanks, guys, for the presentation. And I just had a quick question. Maybe it's a bit fundamental or basic, and I might have missed it. But in the process, as you discussed, and the part of the process is to submit the project to OECD regarding certain testing methodology or testing SOP. And I assume that to carry out this project, of course, the units or institutions that are submitting the project should have sufficient resources to carry out the project, right? So it's not like OECD is supporting the project through the resources, or is that the case?
No, unfortunately, that's not. So the way it works is that countries need to, it's actually one of the requirements when you submit a project proposal is to make sure that you have sufficient resources. So in the past, some of the, for example, one of the cases is that you can be part of, for example, NanoHarmony. A lot of the experts that were involved with us were also part with NanoHarmony because they got funds for doing those tests. We also work a lot. I mean, so it's from our side, it will be if you want on a voluntary basis, but it's really to the leads and to the developers to find the funds. And this is where it's really important to partner with other countries, because then you can divide costs in the production. Yeah.
And is there any in-kind contribution from OECD's, even on the voluntary basis, to guide a consortium submitting the proposal through the process, or that's also not the part of the
the OECD, so in principle, once the project is approved and it comes to our table, then you will be supported by any of us to help you and to provide and to organize the meetings and to give you feedback and to compile comments, etc., etc. We have, I mean, we're not supposed to do it that much in the pre-project proposal, but I think that's, I mean, we have done it anyway. And one way to do it is that countries have come to the Working Party on Manufactured Nanomaterials to share their ideas and to get preliminary feedback and to get interest partners to work together on this. So we do have this, if you want, this preliminary support for those that are interested. Yeah.
Thank you so much. Thanks so much.
You're welcome.
Thank you. I've just got one question in the chat and then one more person with their hand up, Mar.
Okay.
But if we go to Marcus first, and then I'll get the question from the chat. So Marcus.
Hello, thank you. Now it's It's okay. I'm taking part for Vorbiger Consultancy in Freiburg, Germany. I just wonder if there are any regulatory agencies taking part in this workshop, because I think there's the risk of producing a lot of nice in vitro OECD testing guidelines, which finally will not be accepted by ECHA, for instance. And yeah, this would be a pity. So I think it would be very important to involve those agencies in time to result in really useful testing guidelines, which are finally regulatory accepted.
Yeah, so I'm not sure this is for me, but I think there are, but I let Sean to answer that.
Yeah, we do have some regulatory agencies taking part.
Mm-hmm.
And maybe the thing to sort of point out is that OECD test guidelines are really there to address regulation. So there has to, the first thing for a new OECD test guideline is there has to be an identified regulatory need.
Mm-hmm.
Yeah.
So in that instance, in one sense, they are designed to be accepted by regulators.
Yeah.
Thank you.
Right, I've just got one question from the chat. I see two more people have put their hands up. I'll just do the question from the chat first, Mara, if I may, and then we'll make the two people with their hands up. These will be the last two questions asked, because otherwise we will eat into all of our spare time that we've gained. So the question in the chat, Mara, is that OECD methods are developed to characterize and to assess substances. Regulators are also showing an interest on how the substance is and behave in the final product or material where the nano is incorporated. The objective is to better understand what workers are actually exposed. Is OECD the organization that will take care of this too?
So we have several working parties within. I mean, at the OECD, the way we work is that we have a chemicals and biotechnology committee, and then we have official bodies underneath. So the work that I presented today is a working party on manufactured nanomaterials and the test guidelines program. And we have exposure assessment, we have hazard assessment, we have risk management, pesticides, et cetera, Within the WPMN, we are also having projects addressing exposure to workers, consumers, and environmental exposure. And most of the discussion, what we try to do is to have some kind of horizontal discussion when we develop all these methods, is to have this horizontal discussion to make them as useful as possible in all this domain. But yes, I mean, the OECD, if this, again, I mean, we are trying here to respond some regulatory needs to help regulators to implement policies that can provide safety both to human health and environment. So all this will fit within the scope of the OECD.
Thank you. And if we can make the last two questions as short as we can. So Gunther, I believe you were first.
Yes, I will make it short. Mara, I just have a simple question about the graphene-based materials. Which case studies are planned to do with these graphene-based materials?
So we have the first discussion on in December, Gunther, to discuss exactly what will be the scope. I mean, right now, South Africa is doing like a scoping paper if you want to review the different type of graphenes. We have also the documents that was published by ECHA on the different type of graphenes. So what the first discussion is to see what our contributors can provide us with, and to try to frame it a little bit. I mean, as you are already like suggesting, I mean, there are different type of graphenes, and what we want to avoid is to encounter to have something like with the nanocarriers, that we have so many different types of nanocarriers that we cannot come to specific recommendations. So I think that's the first discussion that we will have, and I can keep you updated on what comes out of it.
Okay, thank you.
Thank you so much. And the last question goes to Valerie. And again, short question, short answer, if possible, please.
It's not a question. I just wanted to reply to Marcus about the comment that regulators should be involved in the process and the comment that ECHA should be there as well. I'm now speaking as national coordinator for the EU at the OECD Test Guideline Program, and I can reassure that ECHA is very much involved in the process. I involve them each time, as well as other agencies, by the way. EFSA is also very much involved, and EMA on a case-by-case basis when it's relevant for them. But ECHA is very much involved in the process, and when new project proposals are submitted to the OECD, they are also involved in the commenting of these new project proposals, and they give their advice and comments as well. Just a clarification.
Perfect for that clarification. Thank you ever so much, Valerie. I'd just like to say really thanks to Ma again for joining yet another workshop, as I know we always asked you. And thank you ever so much, everyone, for their questions. I think that was a very interesting discussion, but it's unfortunately now time to move on. And so our next speaker, who I'll hand over to now, if you want to start sharing your slides, is Georges Favre from LNE, who's going to be talking about the European Partnership on Metrology and VAMAS and opportunities for pre-standardization activities. The floor is yours.
Thank you, Sean. So good morning, and hi to everyone. I will try to share my screen, so please tell me if you see my slides, and I will try to have the slide in full screen mode. Sean, can you tell me if it is okay?
It's perfect. Thank you, George.
Okay, great. Perfect. So yes, once again, thanks for giving me the opportunity to give you some insight on VAMAS and the European Partnership on Metrology, because I think that Ma explained very well what OECD is doing to support the development of test guideline, harmonized test methods to support regulation. And I think that there are also complementary initiatives useful to know and to be aware of to support all of this work on pre-standardization of characterization of and testing methods on advanced material to support innovation. And in the next minutes, I will give you some information, so basic information and insight on what VAMAS and European Partnership on Metrology can do on this topic. So we must keep in mind, and I think that I am not going to take anything new from you on this, that in the case of advanced materials, innovative materials, nanomaterials, you can talk about a lot of things regarding those kind of new materials. There can be a regulatory framework in place, or this regulatory frameworks still needs to be developed. There are risk assessment procedures to be implemented to ensure protection of workers, users, and the environment. But most of the time, the starting point is testing data, which is either very scattered, unreliable, and very often difficult to compare from one lab to another. So to be able to move forward in a more relevant way on this, I'm sure that all of you will agree that standardization is key to produce data which can be comparable. But on the other hand, you will have metrology which will make it possible to produce data which are reliable and precise. Of course, these two frameworks complement each other and should be considered together wherever possible. This is an essential precondition for having confidence in data, and therefore in regulation and risk assessment process, which ultimately allows making innovation more readily acceptable. But if we want to make progress on these issues of harmonization of characterization and testing methods, for advanced materials, we need a framework to host initiatives, and at the same time, funding to carry out this necessary pre-standardization work. So OECD WPMN is a good place to develop testing methods to support regulatory requirements. But I think that there is place and complementary places where such kind of work can be made. In particular, regarding initiative, VAMAS is a very good place at international level, with a sort of gateway at the European level through the European Metrology Network on advanced manufacturing recently created. And I will give to you some information about VAMAS and this European Metrology Network in some minutes. Regarding funding issues, which is really key for everyone, it should be borne in mind that European Metrology Partnership coordinated by EURAMET, which is the European Association of National Metrology Institute, offers possibilities for financing this type of research work. So in the next minute, I will give you some information on this program to better understand what the European Partnership Metrology can offer, with specific calls to support pre-standardization issue and R&D. And this program can be a really good tool to feed work within VAMAS or even OECD WPMN. So now, what is VAMAS? Actually, it is the Versailles project on advanced material and standards. And to summarize in a few words what VAMAS offers, it is an international network to set up inter-laboratory comparison to validate measurement methods or SOPs before their standardization. And this network enables also the sharing of good practice regarding measurement and testing on advanced materials. So VAMAS is connected to key bodies and initiatives in this field of standardization and metrology, in particular ISO, IEC, and in the metrology world, the Bureau International des Poids et Mesures, EURAMET at European scale, and the Asia-Pacific Metrology Program, which is a EURAMET counterpart in Asia. When VAMAS has been created, so a long time ago, 40 years, and for those of you who are 40, almost or over 40, you might recognize some of the people in the photo. Actually, VAMAS has been created during the 8th G7 Economic Summit of the GATT, and many projects were funded at those time, and VAMAS is the only one surviving and growing today. And the idea behind VAMAS was to support pre-standardization research to facilitate world trade in new technologies using advanced materials. Once again, the scope of VAMAS is to promote world trade innovation and adoption of advanced materials through international collaboration that provide the technical basis for harmonization of measurement methods, leading to best practice, reference materials, and standards. So at the beginning, the network was created, as I said, in 1982, and was initially limited to a number of countries, which were 7 at that time, namely Canada, France, Germany, Italy, Japan, Great Britain, and the United States. And over the years, the base of countries that officially joined VAMAS expanded. VAMAS celebrated its 40th anniversary last year, and has since produced a large number of standardized testing and characterization methods on a wide variety of topics, actually. There are many working groups, which are called technical work areas within VAMAS, on very different topics. And you see that regarding advanced materials, some of them are of particular interest, I would say. So in particular, regarding surface chemical analysis, polymer nanocomposites, nanoparticle population, Raman spectroscopy and microscopy, and even on graphene and relating 2D materials. What are the key activities of VAMAS? Actually, they can be summarized with this, what you have on screen. So foresighting, in particular, bringing together experts to understand what are the needs of stakeholders and what are the requirements for advanced materials, to facilitate global collaboration by organizing a collaborative project and mainly inter-lab comparison to validate methods, to disseminate best practice, and to access to expertise of specific laboratories across the world, with a common objective to accelerate standardization of testing methods. If you want more information, a good way for that is to have a look to the VAMAS website, and you have the link on the screen. And now, regarding the process to develop a new project, what it is. Actually, the first step consists of filling a form which specifies the measurement issue, the state of maturity of the technique or of the SOP, and if action have been already taken at national level, for example, then after that, to recruit some experts across the world to support potential inter-laboratory comparison, and finally, to share the measurement protocol and the sample to support this inter-laboratory comparison exercise. There are points to consider, in particular, that VAMAS will support work to validate methods when there is a really improvement regarding the measurement approach, and also, and it is important regarding what Mar already explained, to support safety associated to advanced material or nanomaterials. So it is important to have industry and end user participation, and to avoid the duplication of effort already underway in other organizations. So VAMAS is really complementary of OECD, and yeah, I think that close connection needs to be found to avoid such kind of duplication of effort in the future, or to have complementary action, I would say, between both organizations. New project needs to be approved by the working group chair, and new technical work area can be created, can be initiated once it is approved by the VAMAS steering committee. So the benefit to stakeholders, it is in particular to have an insight into new standards for materials, insight into novel material technologies, and access to global network of experts. So it is a good opportunity to define and learn best practice, develop skilled workforce, and benchmark capabilities regarding different kind of measurement techniques, and to help laboratories to compare themselves to expert lab, I would say. And at the end, all of this needs to accelerate the standardization process and facilitate early adoption of advanced materials. Some example in a few seconds. Over the years, or since maybe I think this work has been done 7 or 8 years ago. There was a CODATA VAMAS working group to work on the development of a uniform description system for material on a nanoscale in order to answer to the question how to determine if two nanomaterials are equivalent. So this work led to some recommendation and tools accessible within the CODATA website if you want more information. Some example regarding interlaboratory comparison, and a recent one coordinated by Dano Doraba from BAM, Germany, on the measurement of number concentrations for specific kind of sample using specific techniques. And a recent new project pushed by the Nano-Steffi cluster, if I'm not wrong, to develop line notation and unique identifiers for nanomaterials and groups of nanomaterials, which is really key to support grouping of nanomaterials within rich European regulation. To do the link with what has been said during the presentation of Mar, there is a technical work area dedicated to graphene and related to the materials, and tomorrow you will have more details on what is done within this group through the presentation given by Charles Clifford during the second day of this workshop. So yeah, please, I hope that we will be able to attend tomorrow. Who can participate to VAMAS activities? Actually, any organization with expertise and capability in the area of the study. There is no fee. The participant can be from either VAMAS members or no-member regions, but the participant needs to fund their own involvement in the study. So the test materials for the interlaboratory exercise are supplied free of charge. And if you want more information regarding what is already ongoing on this interlaboratory comparison exercise, please have a look to the VAMAS website, because it lists all the studies ongoing and that are open for participation. Just to do the link with Europe, Euramet, as I said, which is a European Association of National Metrology Institute, launched 4 years ago European Metrology Network with the idea to provide a coordinated offer to meet the needs of EU stakeholders in a more efficient manner from a metrology point of view. So today there are 11 EMN, so European Metrology Network, one being dedicated to advanced manufacturing with a pillar dedicated to advanced materials. So the idea of this European Metrology Network is to facilitate access to EU metrology expertise and infrastructure, and also to help better define the strategy in the field of metrology to support stakeholder needs and the choices that the National Metrology Institute community has to be done, actually. As I said, this European Metrology Network is organized according 3 pillars, one being dedicated to advanced materials, and this EMN enables this year to have a memorandum of understanding signed between VAMAS and Euramet to strengthen pre-normative action in the field of advanced materials at European scale within Euramet, to facilitate the setup of collaboration with key stakeholders, and to move a step forward regarding the identification of priorities. So all of this work through VAMAS and this European Metrology Network is useful and relevant to have projects to be funded in the future through this European Partnership for Metrology, which is a tool supported by the European Commission Eurozone Europe program, focused on the improvement of measurement method techniques, reference materials, and all of this question of measurement to drive innovation, but also to support societal challenges and regulations. It is a really good way to bring together European Metrology Institute, industry, academia, but also standardization and regulators to collaborate on joint research projects, actually GRP. And this program is implemented by Euramet. So if you want more information, please have a look to the website of this EPM, and you have the web link on the slide right now. But what is interesting with this program is that each year you have a specific call dedicated to pre-normative action. And in the next year, a dedicated call restricted to regulation is planned. So I think it is therefore a really good tool to support the development of methods, to support development of methods and harmonized methods linked to regulatory requirements associated to advanced materials. There is a stir-and-peer process between CEN, CENELEC, and Euramet to collect the research needs from the standardization group and to feed this process and the implementation of new calls. And here, OECD can have its say. So actually, there are two kinds of normative projects that can be funded through this pre-normative call. And the project in the strand number 2 is really useful and relevant to address this question of regulation associated to advanced materials. And there is some requirements to lead or to propose some project. Actually, it must include at least 3 national metrology institutes within the consortium. It must be led by a national metrology institute, actually. This year, it was permitted that the project to be led by someone else, that National Metrology Institute. So for the next call, this point is now under consideration. So I don't know what would be the answer from Euramet, but yeah, once again, the main difficulty here is to discuss at early stage with the National Metrology Institute to convince them about interest of having pre-standardization project. This project has a maximum duration of 3 years, and some money is reserved, dedicated to external partners. So you see around 30% of the budget, having a project having a budget about 1.3 million euros approximately. So the process is quite similar each year. So until the end of the year, there is the collection of normative needs. And you see that this year, this needs or this step is expected to finish mid-December. Depending on the needs identified, there is submission of ideas early next year with potential research topic, which are called PRT, and depending on what are the PRT proposed by a small group of partners, there is a selection of base ideas by Euramet, and then call for proposal during the summer to have the final selection of project in November or December to have the project launch during the semester of the year after. So to collect the needs, there is a form available of the CEN-CENELEC website, but it is important to have in mind that all standardization needs will not result in a PRT, so potential research topic. This depends on the Metrology Institute interested, and of course, having this requirement of 3 national metrology institutes working within a consortium. So as I said, this form is available on the Senelec website. You have the web link on the screen. And Euramet recommend to have early exchange between technical committee, working group, and metrology expert to ensure to collect relevant metrology research topic and higher quality for the PRT. So OECD can act as SEN here and express needs. We need to increase the link between Euramet, the metrology community, and OECD expert to facilitate future collaboration and getting opportunities to use such kind of funding to support, to my opinion, test guideline developments in the future. So these standardization needs are published on Euramet website in January for wider dissemination. So if you are interested in, please have a look to the Euramet website in some weeks. And if you want more information on all this process, please contact the person within Euramet responsible for this stir-ampere process, which is Evelyn Domini, and you have the email address of this person on the screen. So just to conclude and to highlight that standardization, pre-standardization activities are really key for the metrology community at European scale. You see that tomorrow in Brussels, there is a meeting open to everyone to discuss the link between metrology, standardization, and certification. And I think that everyone will be within the workshop tomorrow, but it is only, this slide is only here to show you that Euramet wants to support standardization process, regulation, and that this is this EPM is a really good framework to get funding to finance pre-standardization of testing methods, I guess. So to conclude, some acknowledgement to Fernando Castro, the chair of VAMAS, Evelyn Domini, and people behind the European Metrology Network on advanced manufacturing. So thanks a lot. If you have any question, you have my email address on the screen.
Thank you ever so much for that, gorgeous. So we have a first question, in fact. So Blanca, I'll hand the floor to you to ask a question.
Hi, yeah, it's only a clarification. This was really interesting. Thank you so much, George. So from the NIA, we have submitted some standardization needs from our members to SEN. So how do you share this information? Some of those, will they go to you and VAMAS? Or I mean, how is this information shared? It was not clear to me because we channel everything through SEN.
Actually, thank you, Blanca. I don't have any information right now on this, but these needs are collected by Euramet through the stir-ampere process and then published, as I said, on the Euramet website in January. Once this need will be displayed, everyone can propose a potential research topic, which is a 4 or 5 pages document, and it is mandatory to have National Metrology Institute supporting such kind of PRT for sure. So I think that we have more information in some weeks, but if you are share your needs to SEN, I think that the needs will be provided to Euramet to be displayed on the website, and then say, okay, there is a need. Who could be interested to propose a PRT, and then try to have support for this PRT through the National Metrology Institute community. Actually, I would say.
Okay, so we do it once. This is enough. Okay. Thank you so much, George. Thank you.
Are there any further questions? If not, then Georges, thank you ever so much for a very interesting and in-depth presentation. And indeed, I think your slides are something that people probably want to take away and have a look at afterwards. And there's obviously a lot of detail in there. We'll move on straight away, if we may, to our next speaker, who is Valerie Zuang from ECVAM. If you want to start sharing your slides, Valerie, and then I can give a quick introduction. So Valerie will be talking about the contribution of ECVAM towards validation and regulatory acceptance of in vitro methods. And Valerie, the floor is yours.
Do you see my slides?
I do indeed see your slides. You might want to just put them into presentation mode.
I start my video as well. Okay. Okay.
Perfect, thank you.
So good morning, everybody. My presentation will be about UL ECVAM's contribution to validation and regulatory acceptance of test methods and approaches. But first, I briefly introduce the entity where I'm working, namely UL ECVAM, the European Union Reference Laboratory for Alternatives to Animal Testing, which is an integral part of the Joint Research Center of the European Commission. UL ECVAM was formally established under EU Directive 2010/63 on the protection of animals used for scientific purposes, with the following broad tasks and duties: guide and promote research on alternative methods, coordinate validation within the EU, disseminate information on the 3 Rs, which is replacement, reduction, and refinement of animal use, promote stakeholder dialogue, and promote international acceptance. On this slide, you see the life cycle of a new regulatory test method or approach, from research and development to implementation. Research and development of new test methods can take place under various frameworks, such as, for example, EU-funded R&D projects, such as the ones that we are discussing over these 2 days, or national governmental or non-governmental funding schemes, charitable foundations, or industry sponsorships. If methods are then submitted to UL ECVAM for evaluation, the test submitter is invited to fill in a test pre-submission form, which can be retrieved from our website. This is a straightforward, simple form, allowing us to make a preliminary assessment of the status of development, or optimization, or validation of a test method, depending at what moment it is submitted to us, and also of its potential relevance. If the evaluation of the pre-submission is positive, we invite the test submitter for a full test submission, which includes more detailed and comprehensive information on the submitted test. On the basis of the information provided by the test submitter, we assess if the test is ready to enter either validation or peer review, and in the case of validation, what sub-process would be the most appropriate, for example, a prospective validation, or in the case of a similar method, validation based on performance standards. If validation has already been conducted, the validation data and the additional information on the validation study should be provided in the test submission template. You can find all the test submissions that we received so far in our tracking system for alternative methods towards regulatory acceptance, known as with the acronym ZAR. ZAR indicates the stages that methods have reached in terms of acceptance as a recognized test method for use in various sectors, together with a summary description. Where available, ZAR also includes relevant records and documents associated with a method linked to the different steps of the entire process, from submission through validation, peer review, recommendations, and regulatory acceptance, including international standards such as the OECD test guidelines, for example. In the example on this slide, you can see that the 3D reconstructed human skin COMET assay is, for example, currently under peer review.
Valerie, sorry to interrupt you. I think there's just a few problems with people seeing some of your slides.
Oh.
Because you appear to have an issue, I think, with a Zoom side screen, and especially if you go up to the top and look in, you should be able to see what people can see, and there's a grey bar on the right hand side of the screen.
I see it normally on my screen. So, okay, if the regulatory relevance of the submitted test is unclear, we may decide to consult our network of regulators called PARERE, and which stands for Preliminary Assessment of Regulatory Relevance Network. This is a transsectoral network of regulators from the EU member states, relevant commission services, and relevant EU agencies such as ECHA, EFSA, and EMA. It has been established in response to Article 47 of EU Directive 2010/63, which requires that member states shall nominate a single point of contact to provide advice on the regulatory relevance and suitability of alternative approaches proposed for validation. The feedback we receive from PARERE is then usually also transmitted to the test submitter in a summarized form. Now I don't manage to move ahead anymore. Don't know why. Ah, okay. Okay. If the validation of a test method or approach is considered of high priority and the resources are available at UL ECVAM, we may decide to coordinate and carry out a validation study with our network of validation laboratories, which is called EU NetVal. Sorry, I have problems with moving forward with my slides. I think it takes always a bit of time. EU NetVal was set up by UL ECVAM in response to another provision of Directive 2010/63, which requests that EU member states should contribute to the validation of alternative methods and assist the European Commission in identifying and nominating suitable specialized and qualified laboratories to carry out such validation studies. Currently, there is a total of 33 members, of EU NetVal laboratories spread over the EU member states and selected against predefined eligibility criteria. And they are also endorsed by the national contact points for the implementation of EU Directive 2010/63. The financing model employed to support EU NetVal activities will vary in nature, depending on the specific tasks. In general, however, the costs of EU NetVal activities are covered by a combination of direct and indirect financing from member states, EU NetVal members, and the Commission. At the bottom of this slide, I provide the link to our website for more information on EU NetVal, including its terms of reference. Ah, now it's working. Due to the increasing number of developed in vitro test methods and increasing demands for their validation, validation studies are now also often carried out externally, which means not within UL ECVAM, and the full validation data package can then be submitted to us for independent peer review. After our evaluation, independent peer review of the validation study is carried out by the ECVAM Scientific Advisory Committee, known as ESAC. ESAC includes 9 scientists appointed in a personal capacity based on their expertise and selected via a public call for applications. Its mandate is for 5 years, and when needed, the ESAC members are supported by project-specific temporary groups of experts in specific fields. The ESAC acts as a scientific peer review body by providing ECVAM with its opinion on the adequacy and outcome of validation studies, either coordinated by ECVAM or submitted to ECVAM for evaluation. The ESAC may also provide scientific advice on other scientific issues of relevance to the work and mission of ECVAM, as for example, on non-animal derived antibodies or any other topics of high priority. We may decide to issue a new ECVAM recommendation on the validity status of a method or an approach, taking into account the ESAC opinion, our own evaluation, and any other relevant information. The aim of the ECVAM recommendations is to provide our views on the validity of the test method in question, to advise on possible regulatory applicability, limitations, and proper scientific use of the test methods, and also to suggest possible follow-up activities in view of addressing knowledge gaps. We then invite inputs on this draft recommendation from Parere, our network of regulators, ESTAF, which is our stakeholder forum, and our partners from the International Cooperation on Alternative Test Methods, known as ICTEM, as well as comments from the test method submitter in what we call a right to be heard process, and the general public. International adoption can occur in different fora, such as the OECD, ISO, ICH, VICH, or Pharmacopoeia, depending for what purpose and context of use the method has been developed and validated. If the test method is submitted to the OECD, this is done via the national coordinator of the respective OECD member country, or in our case, the national coordinator of the European Commission. If a method or approach is proposed for inclusion in an OECD test guideline, the member country or the European Commission needs to submit a new project proposal to the OECD via his or her national coordinator, provide the validation and peer review outcome, and if the project is approved, draft the test guideline. The draft test guideline will be discussed at an OECD expert group level and undergo several commenting rounds by experts and national coordinators. The final test guideline is approved by the working party of the national coordinators of the OECD test guidelines program, known as WNT, that meets once a year in Paris. In the EU, then, the approved OECD test guidelines are automatically taken up in the EU test method regulation and become official test methods to be used for chemical and product safety testing under REACH and various other regulations, such as, for example, the plant protection products regulation or the biocidal products regulation. As we have heard before, as we have heard already before from Mar, a high level of standardization and acceptance of new methods is indeed achieved by incorporating them into international test guidelines. OECD test guidelines, for example, are covered by mutual acceptance if the studies are generated under good laboratory practices. Regulatory authorities receiving the data under MAD know that high quality and scientific standards were followed, No repetition of the testing is needed for the same data requirement, which increases efficiency by saving time and resources and also thousands of animals. However, ECVAM is also working at other levels of standardization and acceptance of methods. At the level of technical standards, for example, technical specifications defining requirements for products, production processes, services, or test methods have multiple uses and keep pace with new methods development. Technical standards play an important role in innovation. Academic studies use the bespoke tools and design, and they usually tackle complex problems, and best practices there influence the quality of the data. Regarding technical standards, ECVAM is actively contributing to standardization activities of organ-on-a-chip devices for their increased use for internal decision-making and regulatory applications. In 2021, ECVAM investigated the standardization needs for organ-on-a-chip devices, which was followed up by a Putting Science into Standards workshop organized by the JRC and Sensenelec. A Sensenelec focus group was subsequently launched in March 2022 with the aim to map current activities, identify specific issues, draft a roadmap, and define priorities for the next standardization activities on organ-on-a-chip. We also started to collaborate with the European Medicines Agency's working party on the 3Rs on the qualification of organ-on-a-chip. A recent study carried out by Borchert et al. Showed that 58% of key studies used to restrict substances under REACH were non-standard studies. Academic data usually feed into regulatory assessment processes among other data sources, although commonly not conducted according to standardized test guidelines. They may be relevant and complementary to more standardized information requirements. In 2022, UL ECVAM hosted a workshop on improving the use of academic data in regulatory assessments, which discussed and proposed possible solutions to improve current practice. This year, ECVAM also proposed a new project to develop a guidance document to improve the use of academic data in regulatory assessments to the OECD Working Party on Hazard Assessment, which has been accepted on the work plan. So this was a snapshot of our activities at UL ECVAM in this limited time frame. If you want to have a more complete overview of our work, please consult our UL ECVAM status report on non-animal methods in science and regulation. And you can find here the QR code where to download it. Okay, thank you.
Thank you ever so much for a very informative presentation, Valerie. I'll open the floor quickly to questions if anybody has any. No, on that then, thank you ever so much, Valerie, for that presentation. The workshop now sort of moves on and we come to the next speaker. And this is the start of a session of talks on developments of in vitro models and bridging them towards standardization and harmonization. And the first speaker in this section is going to be Tommaso Serchi from LIST, who's going to be talking about MACRAMÉ's approach towards the harmonization of in vitro ex vivo models for inhalation toxicology.
On?
Yeah, please, Tommaso, you may start.
Okay, sorry, I lost you for a moment. Okay, thanks very much. So today I will present you what we do in the MACRAMÉ project about harmonization of in vitro and ex vivo models for inhalation toxicology, not only because we also have a part of Nicotox, but the main focus of the project is on inhalation tox. So just as a reminder for those of you who are not familiar with MACRAMÉ, it is a European project which aims at detecting and characterizing and quantify advanced materials. And we mean really for real life products throughout their life cycle. This includes assessing the potential impact on human and environmental health, and developing characterization methods to demonstrate the effectiveness and efficacy of the method. And again, as I said, applied to market relevant industrial material. And overall, what we hope to achieve by the end of the project is to initiate the standardization, harmonization, technological and regulatory validation of test methods for inhalation toxicology. Now, as an approach, MACRAMÉ is based on real case scenarios. We have 5 demo cases that we do. For each one of those, we have selected and carefully analyzed the different phases of the life cycle, and we have assess exposure point identification and sampling, which are the points where we will take the material from the beginning, different phases of production, use, and end of life, and samples, for assessing if we can characterize them from a physical point of view, and if we can assess the toxicity. All these are information are centralized in the MACRAMÉ data hub, which contains data stewardship, management, and spreading of metadata, reporting templates. We have a SOP recorded and all of that. And then we have 2 big experimental parts, one which is about the development and testing of characterization and PhysChem characterization of the samples, including methodologies for sample prep, imaging, quantitative detection, and whatsoever. And then we have a part on toxicity, where we have some ecotox, a lot of different models for the lung, and overall toxicity testing. All of this is processed together, and then we will aim at producing different data, scientific data, but also standard for submission to ISO, SEN if possible. And overall, we hope to get a kind of guidance documents on in vitro inhalation tox. These are a bit more in detail with different test cases that we have. So we have a different type of advanced materials. They're all based on carbon materials. So they're extremely difficult to detect. We will see what we can do. I heard before there was some question about graphene. We are working on graphene, different type of graphene, which present different difficulties, especially to be detected in biological matrices. Then we have carbon nanotubes, which are taken from car seats, and then we have also nano carriers for drugs. As I mentioned, we did a very large reasoning on where to sample the test cases, and you see we have from pristine material to end of life. We have incineration steps, release from a package, shredding, and other things in between. And you see not all materials will be sampled in every place, because not every place is relevant for them. And we have a part where we sample the test cases for human tox, and a part where we do it for PhysChem characterization, and then also for ecotoxicity testing. Now, when it comes to what we want to do, we have set up a different lines. So we focus for what concern the lung on alveolar region and on bronchial region, and we have developed a tiered approach, going from simpler methods, not entirely simple, but simpler method, to very advanced and complex and expensive ex vivo models from human samples. And I will provide some more details after that. And we also have, as I mentioned, the part on ecotox. I will focus on this at the moment, and then I will not touch it again. So for the ecotoxicology part, we are developing new assay, which are based on fish cell lines, so that we also try to remove and to reduce the use of manual models in ecotox. Then we have the microalgae, and we want to adapt and further test the Daphne assay using these advanced materials. This work will focus on acute toxicities, subchronic toxicity, and chronic toxicity, doing the usual endpoints which are in the guidelines, plus something more. Now, for what concerns the inhalation part, we have divided conceptually the work in different parts. So we try to cover from the very beginning to the very end of the process. And the very beginning is how we generate reproducible aerosols from the materials we have. It might seem simple, but it is actually not. We have many different exposure devices. We have many different ways of producing the aerosols, and they all have advantages and disadvantages. Also, since we have some low-tier approach, which are based on submerged experiments, we want to know how to generate appropriate suspension and how to give it in a good way to the cells. Then the next step is, once you have an aerosol, how you give it to an in vitro biological system and how you determine the delivered dose. Sometimes it is quite straightforward. Sometimes you have to do long dosimetry experiments. Once we have given it to the biological systems, the next step is to understand how predictive are these systems and if information that they give are relevant for regulatory settings. And then once this is done with the standards, how does this setup work with the real life materials? And that's why they have the test cases. I mentioned that we can use different exposure systems, and these are what we have in the consortia, and it is not exhaustive. So there are many more out there. These are what we know best and what are the most common. And we have from a continuous flow, which is probably the most realistic, but also a bit more challenging in terms of time, in terms of materials that you need, in terms of optimization of a generation, also because it varies a lot from sample to sample. Then we have something developed by a company called VitroCell, which is using the vibrating mesh system to generate a cloud, which then settles down on the cells, and it is called the cloud system. It is faster than continuous flow, but it has the big limitation that it doesn't work with dry material. So you have to have the compound that you want to test in a water-based solution, and the same is for material. So it is not compatible with all chemicals and all materials that you want to test. Then we have a newcomer, and it is an instrument that we got as a kind contribution from a company for testing, again, VitroCell, which uses a different nebulization system. So it's a high pressure system. You nebulize the materials that you want to disperse in this cone, yes, that's not a cone, cylinder, and then it settles down on the cells. It is very new, so it has the advantages, but in principle, it is designed to work with a small amounts of dry material or fibers, which would solve the problem of not being able to administer fibers in a fast way, because it is faster than the continuous flow, and we are testing it, so we will see how it works. And then there is the chemical printing, which is based on an instrument from Tecan. It's called Tecan 300D digital dispenser, and there is in fact a printer which prints solution, water-based or DMSO-based, directly on the cells. So here are some of the advantages and disadvantages. Some I already mentioned. For the submerged exposure, it is sometimes not so relevant if you talk about inhalation studies. Nevertheless, the AMA assay is very predictive. It's very useful to have a faster understanding of the toxicity ranking of the different materials. It has a big advantage if you do the AMA assay, but you can deposit the materials prior of adding the biological system. So you can deposit the material on a micro dish and then add the cells afterwards. However, we have to keep in mind that since it is not very simple to create stable dispersion, sometimes you have to add additives which introduce biases and in the readout. And then if a compound is soluble in water, you also start to change very, very quickly the nature of the compound. But will dissolve and not be an nano anymore. The continuous flow is very realistic. It works with dry or wet materials, and it is compatible with whatever you need. So if a material is in solution, it is compatible with water or organic solvent-based solutions. However, it requires large amounts of materials sometimes in terms of grams. The deposition efficiency, and that is why you need so much material, is very, very low. You cannot really make an assumption of the dose, and you have to measure it all the time, and you have to redo the calibration and the measurement for each material that you nebulize. And then also results of what I just mentioned, it is long, the handling, the cleaning, and the exposure takes a lot of time. So it's relatively low throughput. The cloud system, on the contrary, is very high throughput. You can estimate the dose because there is a range of deposition efficiency, but it's better if you check it every time, and you can use quartz microbalances, which is also true for the continuous flow. You need to have water-based solution, which exclude all chemicals and materials which are not water dispersible or water soluble. Also, if you have large materials, you need to assess if they go through the mesh, otherwise you won't nebulize it. It cannot work with dry material, which is a major limitation. The high pressure nebulization, I already mentioned, it is quite quick to perform exposure. You don't need much material. You can only use dry material. And again, you can estimate the dose assuming that all the materials is deposited. The printing, it is very short in handling and cleaning time. It is compatible with both organic and water solution. And you do an absolute dosing because you print and you deliver like pipetting. So everything that you put in there will go there. However, it cannot work with dry material, and only small short. When I prepared the presentation, I wasn't sure. Now I know that it is up to 1 micron that you can use as materials. In order to test all of these and to compare the different exposure systems, because we are going to test all of them and get protocols. We will basically use different materials that we know very well in different doses, and then we will have pictures of the deposited material. We will make the dosing using microbalances if possible. We will assess how much time you need to deposit the material on the cells, and also how you can optimize the handling time, including setup, exposure, and cleaning. In this exercise, we also exchange some limited equipment to see if different labs with the same setup can obtain the same results. In all this, we also test a new instrument which we have developed in LIST together with Vitrocell within another European project. We call it the NebuList, and it is a cloud chamber which has been modified to be faster and a bit more efficient. So the advantages that we expect is to have a higher deposition efficiency, but it is faster and to set up and cleaning. It is a bit more stable because the Vitrocell cloud changes the deposition efficiency by warming up, while this one, it is a controlled temperature. Then with this part here, you achieve a complete separation of the apical part from the basolateral compartment of the cell culture model. And because you work in plates and you don't need to manipulate so often the insert, and you don't need to put them in and out manually, you have a much lower risk of biological contamination and virtually no chemical carryover between exposures. This part here, it is also useful for the tech and digital dispenser because it helps keeping the inserts centered and fixed in the well. Otherwise, they have a tendency to move around, and then the printing is not efficient. When I say printing, it is exactly what I mean. So this instrument here is in fact a printer from HP, where here you have a printing head, which makes nano droplets on the surface. You have to define a pattern, and we have done a long work of optimization of how many dots, where you want to have them. And then we have tested it, and these are the dotted grids using a tracer. As I mentioned, you can work between solution with between 70 and 100% of a DMSO. Nanoparticles in low concentration should be printable up to 1 micron. And this, if you want to use water-based solution, you have to add a surfactant, and these are the surfactants that you can use in a low concentration. So normally this does not impact the cell viability. Now, I have some very preliminary result on the Powder X, which was, this experiment was done at BAWA, and they nebulized the carbon tubes NM401. And what we obtain is that you get a considerable amount of single fibers, between 25 and 50%, but you also have some high aspect ratio fibrous agglomerates and low aspect ratio fibrous agglomerates. Overall, these are promising. It seems that we can use it, at least with some materials. I understood that if you use nanoparticles, smaller, spherical, not so long, it is working even better. So we are confident that this will work just nice. Now, the materials that we will use in the project, we have a very long list. Many of these you already know, because these are the usual suspects that you have in many projects. And it is not by chance that we did that. We want to be able to compare with historical data and what it is already existing in terms of characterization and in terms of biological effects. So the list is very long. We will not be able to test all of those in the advanced system. So we will start first with a alveolar macrophage assay, and then based on the preliminary results from that assay, we will select a restricted number of compounds that we will use for the advanced molecular system. One of these is a tungsten carbide nanoparticles. These are here because they are part of the proposed TG for macronucleus determination. Now, when we go to the biological system, I mentioned several times different tiers. Here is in the left down corner, you see a reminder of a general scheme. And we start from alveolar macrophage assay, which is basically only macrophages from these are murine macrophages. And then based on the inflammatory response and that we get, we can predict if the ranking of the toxicity of the different materials. Then the next tier is going to human cells at very liquid interface using simple models. So nothing too fancy. You have a transfer, you put the alveolar cells or the bronchial cells, you expose them at very liquid interface or in submerged if you want. And then you can assess, again, simple biological endpoints for toxicity. The next tier is already much more complicated. We have the AliSense model, which was developed in LIST. It is based on cell lines, and it is a tetra or pentaculture depending on the need. And you can assess respiratory mutation, synthesization, genotox, and a lot of other endpoints. In parallel to this, we have primary cell-based assays from epithelics, the alveolar model and the mucillar model, which are derived from a human material. You can also have pathological status or healthy status, and they are able to provide a very complex tissue, which is representative of the in vivo situation. As a fourth tier, we have precision cut slices. I have here a picture from a bronchial slice. So you see the different cells in the tissue. You see the cilia. This is the ultrastructure. And you can do much more in-depth mechanism of action studies using these models because they are more representative of a human tissue. The endpoints that we will look at, we have a classical one, including cell viability, cytotoxicity, oxidative stress, and all the rest. Some of which we will try to go a bit farther. So micronuclei, we will also test FACS-based approaches, which are a bit faster and do not require so much handling of the images afterwards. And then for all of these, when we use FACS, we will also optimize the protocol for the dispersion of the cells because you need to have a single cell dispersion. In addition to the, let's say, classical biological endpoint, we're also exploring some more fancy technique. And in particular, we are comparing what you can get from single-cell transcriptomics as compared to bulk transcriptomics. Now, single-cell transcriptomics is known. It's nothing tremendously new, but it, as so far has been very limitedly applied to NAMs. So what we want to understand is if using single-cell transcriptomics, we can get better information and provide better guidance for the improved decision-making process. Now, as compared to bulk sequencing, it is more expensive, it is labor-intensive, but in principle should give you more detailed information at the individual cell level. And so you should be able to distinguish better and analyzing cell type by cell type to also identify more better the mechanism of action. This is what we are doing. We have exposed our system to different chemicals, and these are chemicals which are not materials. We choose these chemicals because we have a long experience in respiratory sensitization and irritation, so we choose compounds that we know quite well, and we have designed an experimental design so that we can compare different settings, different time points, if you need more concentration, less concentration, and this we do for the single cell sequencing and for the bulk sequencing. These are very, very, very preliminary results, which are quite promising. These were our AliSense system, which was exposed to LPS or non-exposed. And you see that you can distinguish very well the population. And you see in the immune cell population, you see very well a clustering of the treated cells against the non-treated cells. While this is not so evident in the epithelial and endothelial cells, where you still see a segregation and a clustering, but it is not so pronounced as for the other cells. And with this, I hope I stayed more or less in the time, and I'm finished.
Tommaso, thank you ever so much for an interesting and good presentation. I will throw the floor open if there's any immediate questions, specifically on the presentation.
Sean, I do have a question, Jacques, but I don't know how to raise my hand.
Okay, Jacques, if we can make questions quick and brief if we can, because obviously we're now standing between people eating. So, Jacques, do you want to ask first, and then we'll move to Blanca and then Rachel?
Many thanks, Sean. Many thanks, Tommaso, for the presentation. It's very, very useful. I have plenty of questions, but one is burning, so I will just stop with one, and I will send you an email for follow-up. Do you plan with these models more the acute tox or the repeated dose tox? And yes, second one, what about the dose? Because you're talking about nasal models, but also lungs model, alveolar models. So the dose at that level will be different. How do you tackle this? And I'm stopping with the questions.
Okay, so for the first question, it's a very complicated question. And I'm not sure we can do everything that we have in mind, but the new trend is to use advanced omics and knowledge graph and another source of information that you combine together, and you analyze the point of departure of the AOP. So everything has to be mechanistically anchored to the AOP. And then we hope that from initial steps, even after 1 or 2 doses maximum, because these models, the ex vivo models and the human primary cells models hold the promise to be able to survive longer in culture. So in that case, with some assumption, you can imagine that you do multiple dosing. Nevertheless, you don't really have a clearance. It is cell culture. So I would be cautious in saying, yes, it survives 2 months, you can expose 60 times. I wouldn't do that. But what we want to do is to try to predict long-term effects by a single dose or a limited number of repetitions. Second question, sorry, was about the dosing. Yes. So for the dosing, it is a bit beyond the scope of MACRAMÉ to do the prediction of the dosing. Normally, what we do is especially if you have environmental material, you estimate the amount that you have. Then you can estimate how much. There are models to estimate how much you inhale, and of this part, how much it is the deposited dose. We did in the past, now it starts to be almost 10 years ago, some studies using a worst case scenario, and we could detect signals which were point of departure for long-term toxicity. So we are confident that even if you use realistic low doses, you still see something that it is interesting.
Okay, thank you for that. Blanca's kindly said that she'll ask her question later, Tommaso, anyway, so we can move straight on to Rachel. And again, hopefully short question, short answer.
Oh, hi, really interesting presentation, Tommaso. Just a few real comments. I noticed you mentioned for your systems you were going to use clean air controls. Can I encourage you to do some incubator controls, in particular for those that have any flow, if you haven't already planned to do that? Because
no, we always do.
Yeah, that's okay. And also, I just wanted to ask you about what your plan, if you have any plans to do some comparisons, because we compared using a silver nano aerosol in vivo with in vitro using a Cultex system, and it was quite difficult to try and match them. So I wonder if that's maybe beyond the scope. And a final point really is looking at longer term effects. The effects, it's very difficult to capture using in vitro systems, the effect of the influx of neutrophils if you have a large enough dose. So I wonder if that's a limitation of all these models, but maybe too many questions, Tommaso, and we have to.
I can try to answer quickly.
Have to share email on that.
Very quickly.
Thank you.
For the incubator control, now I'm not privy to all details, but I know that we have clean air exposed, and then there is always some insert in the incubator just to make sure that everything is fine. About the infiltration of neutrophils, I'm not sure, because we do have the insert and we can measure the we allow the passage of the cells up and down. Now they are all there, so they're not really able to go anywhere. It's not that there is a reservoir of cells where they stay, and then we can definitely check the markers which are promoting the infiltration. That we can do. I'm not sure to which extent. We haven't really looked at it. And then, sorry, the last one was about.
That was comparing in vivo with in vitro,
absolutely.
whether that's within the scope.
It is always within the scope, in the sense that that is what we want to do. So to have a fantastic in vitro model, very reproducible but not predictive, doesn't really help us. So it is complicated to do, especially with nano, because the amount of data which are often available for in vivo experiments from nano are different, also because it's difficult to get always the same materials. But we try always as much as possible to link it to real data. Not that our data is not real, but I mean from in vivo data, and from also epidemiological data. So there are certain things that we know how it should be, and we always try to find the link to what we see and what we should see. Now, unfortunately, that goes well beyond oxidative stress measurement. Even some cytokine are all indication. What we need really to have is a solid AOP with molecularly initiating events that we can measure, and that can give an answer and a prediction. Other than that, it is just as saying a ranking of these is possibly more toxic than the other one.
Thanks very much. It's fascinating. I'll try and keep up with this interesting project. Thanks, Tommaso.
Perfect, thank you. I'm going to be a bit of a tough chair, Marcus, and so I'm sorry. If you could save your question, you'll have an opportunity later, as Tommaso will be on the round table panel, so you'll be able to ask your question then. I'd just like to round up this morning's sessions, or this morning if you're in Europe, and say thank you ever so much to all of our speakers. I think we've had some very interesting presentations, and I hope this will stimulate some further discussion later on in the roundtable session. For now, we will take a 30-minute break, and so we'll come back in 30 minutes, which will be 12:40 CET, or 11:40 if you're on UK time. In fact, 6:40 if you're East Coast US, and indeed 8:40 PM if you're on Japan time. But see you all in half an hour. We'll stop the recording during the lunch session. Feel free to leave yourself in the meeting if you want, and we'll come back later on. And for those who asked about Australia, I'm not sure I'd have to calculate that one. Thanks ever so much, and see everybody in half an hour. Thank you. Bye-bye.